Section 1. Trace evidence, hair, fiber, glass, paint, and soil. Trace evidence is evidence that is found in small quantities. This means the size of the substance found is small when looking at it with the naked eye, not that there is necessarily a small amount of evidence. Trace evidence may help investigators tie someone or multiple people to a crime scene, but it can also show deliberate detachment. Trace evidence is circumstantial evidence. This can point investigators in the right direction, but it is not direct evidence on its own. One of the ways trace evidence is examined is through a technique called microscopy. Biological microscopes, stereoscopic binoculars, polarized light microscopes, and comparison microscopes are some of the devices used to analyze different types of trace evidence. Biological samples most often are analyzed through biological microscope or stereoscopic binoculars. Polarized light microscopes are similar to biological microscopes, but they're more sophisticated. They measure the optical and physical properties of the sample, and the comparison microscope is used to analyze the sample side by side. Hair is made up of layers. The outer layer is called the cuticle and is made up of keratin scales. This makes the hair tough and unaffected by chemical action. Hair dye shows on the cuticle cells but does not seep down to the second layer known as the cortex. Keratin scales from hair on animals is seen in a pattern under a microscope, and for humans the keratin scales have no pattern. Human hair also has an irregular shape to it, and animal hair has more uniform shape to it. The cortex cells are called cortical cells. They are shaped like an hourglass and have no pigment. They make up the second layer. The third layer is called the medulla, and in animals it runs the entire length of the hair, but in humans it can be broken up, appear irregularly broken, or may not even be there at all. Hair evidence should be collected from the crime scene and be labeled unknown origin. At a separate location, hair should be collected from the victim and suspect as well to be used for comparison. All hair samples should be stored in separate paper envelopes to avoid contamination. Hair that has a root will have skin cells joined to it. These skin cells hold mitochondrial DNA. Mitochondrial DNA is DNA from the mother's side. There are natural fibers and human-made fibers. They are identified by two separate classes. Natural fibers are created by interactions with the atmosphere. Human-made fibers, often using cellulose, which is a naturally occurring polymer. Manufacturers have the ability to change the shape of the fibers as they need, so this can be helpful to recognize one from the others with the same chemical compounds. When collecting fibers, sometimes it might be smart to take the larger item it is attached to in order to not damage or contaminate the evidence. Fibers should be stored in a paper envelope and then placed in Ziploc type bags. The polarized light microscope will be used when analyzing fibers because it has the capability to measure the optical and physical properties of the fiber. Glass composition is considered amorphous, meaning it has no clear defined shape. The molecules and atoms have no set arrangement since glass is silicon oxide, heated up until it melts. There are many types of glass, but a few are more commonly seen as in evidence. When collecting glass, it should be placed in paper envelopes or glass bags, then placed in a cardboard box to prevent damage. Sometimes glass needs to be reassembled if a fracture match is requested. All the glass should be collected. This is done with untempered glass only. Glass samples can be analyzed for surface features, color, thickness, and emission of light. Glass can be analyzed by heating it to a melting point, plasma analysis with mass spectrometry, energy dispersive X-rays, and atomic spectrometry. Elemental analysis will not tell you where the glass is from, and doing these tests destroy the sample. Paint contains organic and inorganic chemicals. Analyzing may let you know the manufacturer and exact color number / name. There are 4 coats of paint: the electro coat, primer, the real color, the final coat, which is a protective coat. The International Forensic Automotive Paint Data Query Database is used to find the exact manufacturer, model, color, and years the manufacturer used that exact color. However, if a vehicle has been repainted, it may not be able to be found in the database. Paint scraping should go all the way down to the base in order to get all the layers present. These layers may be analyzed one at a time to check the position of the layer. Spectrophotometry is used to check infrared frequencies absorbed, and for composition, X-ray fluorescence is used. Paint samples should be stored in paper or plastic, and they need to be resealable containers. Soil is any decomposed material that lives in a certain area. It can be made up of anything. Soil can be found on anything: shoes, clothing, tire tread, tools, just to name a few. It should be collected and stored in paper envelopes or cardboard boxes that are sealable, metal containers, or glass vials. Soil can be dried and compared. It can be separated, measured, weighed, and observed macroscopically. Under a microscope, you might find parts of a plant, hair, waste, and it can be tested for minerals or diatoms. The probability, if it is from another area that has similar characteristics, or is this from the crime scene, will be determined at this time. Thank you for listening to my presentation. I got all of our information from the Introductory to Forensic Science by Gienslin and Larson, 2019. I got all my images from Google Images. Thank you.